Thank you very much, Daniela and Morton and Alex for the invitation. Great to be back here. So as you all know, we live on a planet with a 24-hour environmental cycle, and organisms all evolved internal circadian clocks. Is this? Okay. And which look like this. This is a very simple diagram of the core mechanism of the circadian clock in mammals. It involves two transcription factors, clock and BML, that my lab discovered many years ago, that drive two sets of repressor proteins, the period and cryptochrome proteins. These are transcribed and translated in the daytime. They interact with each other in the afternoon. They go back into the nucleus at night and repress their own activators, clock and BML, to form this 24-hour autoregulatory cycle. Just one highlight about what the clock is doing. If you look at the mouse liver and profile transcription and associated chromatin marks associated with Pol2 transcription, what you see is an incredible stereotype transcriptional profile that happens every day in the mouse liver. In the morning, you get occupancy of the activators, clock and BML. Here, binding in the morning. This is followed by bursts of transcription, Pol2 occupancy, and then you get the classic repression phase with the PER and CRY proteins. They're rapidly turned over, and then you go into the next cycle. What's remarkable about this is, there are 3,000 genes at the mRNA level cycling, but at the chromatin level, there are thousands, 5 to 8 to 9,000 chromatin marks that are cycling in the genome every day. And so the clock is really doing a lot to transcription in the mouse liver. This is not a unique picture to the mouse. You'll see this in the pancreas too. But just to give you a flavor of what the clock is doing, if we look at the target genes for this core transcriptional profile, here's the KEGG metabolic chart. In red are shown all the BML target genes. The entire chart is covered, okay? This picture is not unique to the liver. You'll see it in the pancreas, muscle, and in brain. So the clock is really fine-tuning metabolism on a daily basis in a cell-autonomous way. And I think it's one of the most important functions of the clock. Now, we all know that caloric restriction is one of the most important interventions for extending lifespan. But one of the interesting features to us was the classic experiments were done in a funny way. They're fed on Monday, Wednesday, Friday morning. Mice eat every day, not every other day, and they don't skip weekends. And in addition, they like to eat at night, not in the morning. And we know that daytime feeding has effects on peripheral circadian organs. So to look into the effects of caloric restriction more carefully, Vicky Acosta-Rodriguez, who's now with Rafa's group at the NIA, and our lab, developed an automated feeding system which allows us to control access to food, but importantly also measures the timing of the food, and we can then program in any pattern of food availability for the mouse. So this is what it looks like if you plot an actogram. Each line is a day, mouse is night active, shown in black. It's running on a wheel, as you can see here, for 43 days. And each dot is when it takes a food pellet. It takes 14 a day, 75% at night, 25% in the daytime, okay? Now, when we put mice on caloric restriction, a strange thing happens. Their eating pattern doesn't just decrease uniformly. What they do is they eat the food as soon as it's available. So here, the mice can get access at 0, dawn. They eat all the food in 2 hours because they're hungry. Okay, same thing happens at night. And so what this means is when you reduce calories, you're actually changing the feeding behavior of the mouse. It goes into extreme time-restricted feeding, and it has a very long fasting interval. And so we wondered what the contributions of these three factors were to lifespan. So we have the three groups I just showed you, ad-lib feeding, the ones that eat in 2 hours, day or night. Then to get around binge eating, we spread out the food over 12 hours, day or night. Or importantly, we have this fifth group that has continuous feeding, no day-night difference, no fasting. Okay? The mice are active for their lifespan. We have food and activity for every single mouse. And importantly, the feeding pattern is also stable for their entire lifetime in this experiment. And we know exactly what they're eating and when they're eating it. Okay, so here's the body weight. Ad-libs gain weight as you'd expect and then spontaneously lose it after about 20 months, despite the fact that they eat even more food, as Rafa mentioned to us a number of days ago. And the calorically restricted groups all maintain low, healthy body weight. Now here's the first surprise, and that is the mice that ate continuously only lived 10% longer, even though they were calorically restricted 30 to 40%, okay? The mice that eat at the wrong time live 20% longer, so you get a little boost, 10 more percent with the fasting interval. There's no difference between 12-hour fasting and 22 hours of fasting. And then finally, the mice that eat at the right time live 35% longer. So there's a huge effect of the pattern of eating under caloric restriction on the magnitude of lifespan extension. And this was actually surprising to us that the phase, the timing of eating would have actually the major effect in lifespan extension. The mice shown in other experiments die from blood cancers and liver cancer, as we'd expect. Liver has the most lesions. I'll just repeat what we published 2 years ago. If you look at ad-lib mice at two ages, 6 and 19 months of age, 2,000 genes go up, 5,000 genes go down. All the genes that go up have to do with inflammation, as we've seen across this entire meeting, as Deep showed also. All the genes that go down have to do with metabolic pathways. And caloric restriction rescues the majority of these genes. Any form of caloric restriction rescues the same category of genes. And there's a correlation between the extent of rescue of the deviation in gene expression and the lifespan that we see. Okay? So these are rescued by any form of caloric restriction. Fasting rescues another set, new go term, thermogenesis. And circadian alignment then goes back to innate and adaptive immune responses, with interleukin-1 being perfectly correlated with lifespan. Okay, the other feature we find is that the amplitude of circadian gene expression declines with age. So you can see these 5 examples here. Red is the old, young in black, but genome-wide amplitude is significantly decreased. And when the mice are old, if you give them daytime feeding, this totally disrupts their circadian pattern. And so, for example, here, in the mice that eat at night, in 2 hours, they have very nice circadian rhythms that are rescued with age. But the ones that eat in the daytime only have 7 genes that cycle under very strict criteria. So the time of eating is actually very important. It also shifts the phase of genes shown in yellow that are cycling, okay, to the wrong phase. And in addition, if it doesn't change the level, we also find that daytime feeding, shown here in yellow and orange, disrupts the pattern of genes that normally are rhythmic, okay, including some of our favorite longevity genes, FOXO3, IGF-1, which are rhythmic. Okay, so circadian amplitude decreases with age. Caloric restriction can rescue that amplitude. And importantly, the majority of the classic aging and longevity pathways, such as IGF-1 signaling, mTOR, AMPK, SIRT1, PGC1 alpha, those are all direct targets of clock BMAL, direct transcriptional targets. And if we look at a number of metabolic tissues, we see that their gene expression is cycling in at least one of those tissues, and in fact, the majority of them is shown in purple. So the clock really, we think, is a major controller of many metabolic pathways involved in aging and longevity. And in Vera's beautiful paper published a while ago that she showed us, incredibly, the two most conserved modules in this gene expression, comparative gene expression analysis, were circadian clock genes and reprogramming. Thank you, Vera. And then, as we heard from Deep at the beginning of this meeting, in the calorie study, the top hit in adipose tissue under caloric restriction, again, is clock BMAL as a go category. And in another study from Luigi Ferrucci at NIA using muscle biopsies, okay, circadian clocks are a top category that emerges out of this gene expression under caloric restriction. So really, our idea is that the majority of the hallmark of aging pathways are actually strongly influenced by the circadian clock system. Almost all of them actually have direct transcriptional regulation by clock BMAL. So how can we get at this? So we know that clock and BMAL loss of function mutations have a very severe effect on survival, as Pura showed us today in the BMAL knockout. Okay? Also shown by Kondratov a long time ago. And in addition, loss of function of the clock gene reduces lifespan, also shown by Roman Kondratov's lab. Either the mutation of clock or deletion of BMAL in metabolic tissues also has very strong metabolic phenotypes. So in the clock mutant mouse, we saw loss of both lipid and glucose metabolism. And in conditional deletion of BMAL only in pancreatic islets, this leads to loss of glucose regulation, shown here, which is caused by insufficient insulin production. So given that, deletion of clock genes shortens lifespan and has metabolic defects associated with longevity, and that circadian amplitude decreases with age, is there a way that we could enhance or strengthen the circadian clock and restore that amplitude, the robustness of the system, and the resilience in downstream pathways? And we think that that could then promote healthy aging. Okay, so a long time ago, in order to clone the clock gene, we made a back transgenic mouse to rescue the original mutant to prove that that was the gene. Okay? And this mouse, thankfully, is still with us. And it's very interesting because it only has about a 2-fold increase in transcription. But it's global, and its regulation looks normal. And so what we're testing now, and this is being led by Nathan Skinner, who's somewhere in the audience here, is to really ask, does increase in clock gene expression using a transgenic mouse prevent the decline in transcriptional drive that occurs with age? And will then this have health and lifespan benefits, okay? So here are the body weight curves. We already had a surprising result here. This is on regular lab chow, not high-fat diet or anything. The transgenic mice all have lower body weights, males and females, yet they eat more. So we like to do that, right? They also have no significant decrease in energy expenditure. So that's a little bit curious. So we're still trying to understand how they can defend themselves against diet when they eat more and still lose weight. The mice also are resilient to high-fat diet or diet-induced obesity. So these mice are significantly lower body weight, weight gain, or body fat mass, depending on how you want to measure it. And in addition, they also have no significant difference in energy expenditure or RER. In this experiment, we transitioned from normal diet to high fat over this time period. And you can see the high fat diet effect here on the RER in this group, okay? But again, no difference due to the genotype, which is interesting. So, and if we do ANCOVA analysis, there's also no significant difference, okay? All right, so here's the important experiment, survival. It's still ongoing, but what we are seeing is that in males, there's going to be a significant extension of lifespan just by increasing clock gene expression under ad lib feeding, no restriction at all, okay? Unfortunately, the females don't have such a large effect. It is significant, but it's a very small benefit. So we don't understand that either. So I apologize to half the audience for that result. And maybe we can figure it out. And so we consider that a genetic proof of principle that boosting the clock may be beneficial. So can we really find small molecules or drugs that can enhance clocks? And then we can try to use those as agents to boost the strength of the clock, and maybe they'll have therapeutic effects. So the clock, has been targeted by many, including my lab. We saw the use of one of these drugs on the reverb agonist by Eveline Bischoff earlier in the meeting. That's right here. But interestingly, all the existing compounds, with one exception, have all hit what we call the periphery, the repressors. None of them actually are good, have hit clock or BMAL, which is really the driving target or transcriptional gene that controls the entire network. And so we're really focused now on target-based screening to try to find compounds that can either inhibit or enhance clock transcription. And we're in the middle of doing many different assays to try to optimize for high throughput screening. And so we hope to have some interesting compounds for you in the near future. Okay, I'll stop there. This was really led by Vicky Acosta-Rodriguez, incredible postdoc who's now at the NIA, and then I have a huge number of really talented postdocs in the lab working on various aspects of this future work. Thanks.
Thank you very much. Thanks. Maybe we have time for a very quick question.
Thank you. As you know, you can manipulate circadian clock through the light coming to eyes to suprachiasmatic nucleus. So do you think any solution like using light to enhance those circadian clocks?
Yeah, so I mean, being exposed to a strong light signal every day, going outside, being inside is not strong enough really, will strengthen your clocks. I'm not sure that's going to be sufficient, though, by itself. I think you're going to have that plus nutritional signals under regulation.
Okay, one more question there. Rolin?
Joe, that was beautiful work. Do you know why the females wild types are living less than the male wild types?
Why the females?
Wild types in the experiment are living less than the male wild types. Normally the B6s, the lifespan of both are kind of the same. I'm assuming that they are the offspring of the head-to-head cross. So it could be very
so
interesting.
they are litter mates, so
Yeah.
it's not that. And I think it's because all of our mice are singly housed, and thermogenesis is a much larger stress for female mice than male mice. And so most females, are used to living together. That's my suspicion in why we're seeing this, because they're singly housed.
That will be very interesting to look at the fat metabolism, because that is in previous work that we have done, even at 20%. We see that the preservation of the effect of CR is totally dependent on the amount of fat
I
they're
see.
able to keep between year 1 and year 2. It'll be super
okay.
interesting to see. And
Yeah.
that was 30% CR or was?
It was 30% CR.
Okay.
Yeah.
Perfect.
Well, no, I didn't show a CR with the females.
Okay.
That was just just
plain
ad
vanilla
lib
expression.
for the wild types. Yeah,
Thank you.
thank you.